Yes. Ah, I see. Glad somebody's using it. So aerobic respiration. Yesterday we talked about anaerobic respiration, how when we go through glycolysis, the absence or presence of oxygen determines which direction we go, anaerobic or aerobic. So yesterday, anaerobic, no oxygen, we go through alcoholic or lactic acid fermentation, depending on the organism. I'm not going to go too deep into fermentation. You don't even have to make a model of that. I just wanted to let you guys know that there is a process that happens where we don't need oxygen that will help our organisms that don't need oxygen to create our ATP. So today we're getting into aerobic respiration when there is oxygen present. So pretty quick before we get into new stuff, we're going to review glycolysis. So glycolysis happens for all types of cells, prokaryotes and eukaryotes. So this is happening in our cytosol, so outside of our organelles, but still inside the cell, that watery, gooey substance that holds all of our organelles in place. And it doesn't matter if there's oxygen present. Oxygen is not one of the reactants of our glycolysis. So we start out with a glucose up at the top. Let me get my pointer. Up at the top here, we have our C6 molecule. Glucose has 6 carbons, so we're just going to call it C6. And we're splitting it in half. To do that splitting, we need to use 2 ATP. Bummer. We want that ATP for energy, but got to spend some to make some. After that C6 is split into 2 3-carbon molecules, they're going to get rearranged. So we're not adding or removing anything. We're just switching some atoms around in this molecule to create a new thing called pyruvate. During this rearrangement process, we're creating also some NADH, which is an electron carrier, and some ATP. Hey, we got our ATP back. We're going to have a net gain of 2 ATP. We lost 2 at the beginning, and we're ending up with 4 at the end, so our net gain is 2 there. And between these 2 C3 molecules, we're doing this process twice because there are 2 of these molecules. We get 2 pyruvates, 2 NADH, and 4 ATP at the end. All right, so we made our pyruvate at the end of our glycolysis. Now we have to do something called oxidation. So we did our glycolysis, made our pyruvate outside of our mitochondria. If there's oxygen, we're going to move into the mitochondria. We're moving on to our pyruvate oxidation and Krebs cycle. I like to link them together because they are very closely related. Before we get into that, we need to know a little bit about mitochondria anatomy. So looking at our mitochondria, we have this outer membrane holding all of the good stuff inside. It creates, its own organelle, so it needs a membrane to separate it from the other stuff in the cell. And on the inside, we have another membrane called the inner membrane that's holding in this stuff called the matrix. So inside this inner membrane is the matrix of our mitochondria. These 2 things, the inner membrane and the matrix, these are where all of our aerobic respiration processes are happening. So looking again at this, we have at the top this cool actual real life image of a mitochondria. We can see all of the little wiggly things. And then on the bottom here, we have our representation or digital drawing. But we're really going to focus on this inner membrane that surrounds the matrix and the matrix. So an overview of this pyruvate oxidation. We just made our pyruvate, like I just said. At the end of our glycolysis, there's still some energy in that molecule. We want to take it. The whole point of this process is to make as much energy as possible. So we want that energy. So we're going to move that pyruvate into our mitochondrial matrix and create a new thing called acetyl-CoA. We're just going to call it CoA, coenzyme A. That's all you need to know is CoA. When we make this, we're releasing carbon dioxide and making another NADH, which is an electron carrier. So let's look a little bit closer. 3 steps here. We start with our pyruvate that we made that was a product of our glycolysis. It's going to release CO2. So we're getting rid of 2 of these oxygens and 1 of the carbons. That will be waste, our CO2. We're creating an NADH, an electron carrier, and what's left over is called COA. We get 2 CO2s, 2 COAs, and 2 NADHs because there are 2 pyruvate molecules. So we're doing this process 2 times. Here's a different look so we can count our matter, make sure we're conserving our matter. So here is our pyruvate. We have 3 carbons, 3 oxygens. There's some hydrogens over here, but we're not going to worry about them. They're not going anywhere. So we have 3 carbons. Boom, it's going through this protein. Once it's inside the mitochondria, we can do this process. We're creating our electron carrier by sticking a hydrogen ion onto our NADH. We're losing 1 of these carbons and 2 of these oxygens. Boom, carbon dioxide, we don't want you. Bye, see you later. And the result, these 2 carbons left over, 1 oxygen, those hydrogens are also going along with them, but we're not counting those, they're staying. This is our result, COA. 2 carbons, 1 oxygen. And then we have our hydrogens that didn't go anywhere. So here is a summary. It's a very short process. If there is oxygen, we're going to move on to pyruvate oxidation after glycolysis. 2 carbons are released as carbon dioxide. 2 molecules of COA are created from our 2 molecules of pyruvate. And 2 NADH are generated from our NAD+. Everything is 2. We're doing this process 2 times because we started with 2 pyruvate molecules at the end of glycolysis. That's why there's 2's in front of everything. Then these pyruvate broken down molecules, COA, are going straight into the next process called the citric acid cycle, also known as the Krebs cycle. It's called the Krebs cycle for the same reason that the Calvin cycle is called the Calvin cycle. Some scientist dude named Krebs discovered this. As scientists are wont to do, they name things after themselves after they discover them. So it can be called either thing. So know that citric acid cycle is the same thing as the Krebs cycle. Here's a brief overview. So here at the top, because pyruvate oxidation goes straight into the Krebs cycle, a lot of times they're pictured together. So up here at the top, this is our pyruvate oxidation. It's happening and feeding right into our Krebs cycle, which we're going to talk about next. So the first step, so we did our pyruvate oxidation, we ended up with our COA, 2 carbons. Those 2 carbons are going to join up with the leftovers from the previous cycle, a 4-carbon compound. Sound familiar? Should be kind of reminiscent of our Calvin cycle. We're moving carbons around, recycling things. In the Calvin cycle, we would be doing 5 C molecules of RUBP, but since we're in cellular respiration, things are a little bit different. So we're starting with a 4-carbon compound instead of a 5. So this 4-carbon compound joins up with our COA, which is 2 carbons. Putting those together, we get 6 carbons. That 6-carbon molecule is called citric acid. Hey, maybe that's why it's called the citric acid cycle. The next step, we're removing 1 of those carbons through CO2. We don't need CO2. That's our exhale waste. Bye, see you later. And in that process, we're creating another NADH, getting some electron carriers. We're left over with 5 carbons. So we got rid of 1 through carbon dioxide and creating this NADH. There are 5 carbons left in our molecule. We're going to do that 1 more time. We're going to remove 1 of those carbons. Bye. That's a CO2, gone, exhaled. We don't need it. We're creating an NADH again. And this time, hey, we get to make an ATP, energy. That's what we want. So now we got rid of 1 from our 5. We're down to 4-carbon compound. This last step, very, again, reminiscent of our Calvin cycle. We're going to rearrange. We're not going to add or lose anything. We're just going to rearrange this 4-carbon compound. In that process, we're creating yet another NADH and another type of electron carrier called FADH2. So everything in an orange box here on the bottom part, the NADHs and the FADH2, those are our electron carriers. So when we tack on that hydrogen, if you remember, it's trapping in some electrons, and those electron carriers are going to take it over to the next step in our aerobic respiration. Okay, electron carriers. Krebs cycle produces a lot of them. So many. We're making 3 NADHs and an FADH2. That's 4 per cycle. These are going to go onto our electron transport chain. That's our next step in aerobic respiration. This is happening 2 times for every 1 glucose. Start off with 1 glucose, we end up with 2 pyruvate. 2 pyruvate turn into 2 COAs. 2 COAs, they need to go through the cycle 1 at a time, so it needs to happen 2 times. So for every 1 glucose, we're running the Krebs cycle 2 times. So for every 1 glucose, we're getting 8 electron carriers. It's a lot of electrons. So let's put it all together. Pyruvate breaks down into COA. Those carbons are conserved by creating carbon dioxide that gets exhaled. We don't need that. The COA then moves into the Krebs cycle, where it joins up with the remnants of the past cycle, a 4C compound, creating a 6C compound called citric acid. We're going to leave, or rather get rid of one of those carbons through carbon dioxide so that we can make an NADH. We have 5 left. We're going to do that again. We're going to get rid of one of those carbons through carbon dioxide to create an NADH. We're also in this middle step, creating an ATP. Love it. Got to make that energy somehow. And we're left with this 4-carbon compound. We're going to rearrange it so that it can accept the COA in the next step at the beginning of the new cycle. In this rearrangement process, we're creating more electron carriers, another NADH and an FADH2. Again, it's kind of reminiscent of the Calvin cycle. We're rearranging, moving around some carbons, losing some carbons to carbon dioxide. So, there's sort of that tie in there. If you notice, it's a cycle, so things are getting reused and stuff, but the main point is we're creating these electron carriers. So let's count our carbons. So for every 1 cycle of the Krebs cycle, we're starting out with our pyruvate, which is 3 carbons. We're losing one of those to carbon dioxide when we oxidize it, creating our COA. That COA combines with the leftover 4C from the previous cycle. 6Cs, we lose one of them to carbon dioxide, left with 5, lose one of those to carbon dioxide, left with 4. 4 gets rearranged, starts all over again. Again, this is happening twice. We made 2 pyruvates from 1 glucose. So for every 1 pyruvate, we have to do 1 cycle. So 2 cycles per glucose. Looking at our electron carriers, here's what's happening. Our pyruvate becomes COA. COA joins up with the previous the previous remnants of the last cycle, 4C, to create a citric acid 6C molecule. We lose one of those to create our NADH, lose another carbon to make a second NADH and an ATP. Those 4 leftovers rearrange to create a third NADH and 1 FADH2. So for every 1 pyruvate, we're making 4 electron carriers, happening 2 times. So for 1 glucose molecule, we get 8 of these electron carriers. All right, so we made all these electron carriers. Who cares? What's the point? We started with this huge glucose molecule. We made carbon dioxide and a couple ATPs. We just made like 4 ATPs. And a bunch of these electron carriers. Who cares? Who cares? All right, so here's a summary. If you're doing the notes, this would be a good time to look up, fill in some gaps. So the COA from pyruvate, I know I'm going to say it again, but repetition is the key here. So the COA from pyruvate oxidation joins in with the 4C left over from the previous Krebs cycle to create a 6C molecule. That 6C molecule breaks down. We lose one of those carbons to carbon dioxide when we're making our NADH. We're going to do that again. Take that 5C molecule, remove 1 carbon for carbon dioxide, making NADH and 1 ATP. Then we're left with 4 Cs. We're going to rearrange those, make 2 more electron carriers, another NADH and an FADH2. And then that 4C molecule, after it's been rearranged, gets recycled. We're going to use it again to start the Krebs cycle from the beginning again, to join with another CoA from that pyruvate oxidation. All right, so we're counting these ATP. We got 2 from glycolysis. We got 2 from the Krebs cycle, because remember, we're doing the Krebs cycle 2 times, so 1 ATP per round here. So we have 4 ATP. That's not enough. Might be enough for some, bacteria, but we're eukaryotes. We need that energy. We have a lot of complex things to do. We're big. We need a lot of energy. So here we go. Those electron carriers are taking those electrons to the electron transport chain. This should sound familiar. We had an ETC in our photosynthesis. So here we go. Why do we care about these electron carriers? Because they're going to bring those electrons to our electron transport chain. The electron transport chain is a series of proteins in the inner mitochondrial membrane. So thinking about the anatomy of that mitochondria, there's the matrix, and then that inner membrane that holds all the matrix together. So that weird wavy line, wiggly part, that's the matrix. The inner membrane is holding that matrix together. We're going to make a ton of ATP. This is where the magic happens. This is why the oxygen is important. So here, just again, the membrane is the part on the outside of the wigglies, which is the matrix, the wiggly part. Here's a brief overview. We'll break it down, don't worry. But we're taking these electron carriers, breaking them apart to release the electrons. We're using those hydrogen ions that we're breaking off to increase the gradient and ultimately power ATP synthase. Again, this should sound familiar. A very similar thing is happening during photosynthesis. So let's break this down. Step 1, we have our electron carriers that we just made a ton of in the Krebs cycle. They're floating over to the ETC over here. They know they need to do their job. So these proteins that are in the membrane, they're going to break off that hydrogen to release the electrons. That hydrogen is going to float off into the matrix, probably be sucked up in the next step. But that hydrogen is not important to the electron transport chain at this moment. We're gonna get those electrons so we can do some work. Those electrons have a little bit of energy. These proteins are going to use that little bit of energy as they pass those electrons across to do some of this work. What work? Well, we want to increase our concentration gradient. Got to get those H+ ions inside the membrane. This one is upside down, this diagram. The inside of the membrane is on the top, outside of the membrane is on the bottom. So we're pumping. We're using the energy from these electrons to get these hydrogen ions into the membrane. Got to build that concentration gradient. If you remember from photosynthesis, it's because we need to power something at the end here. So before we get there, last step, the last protein in this line of the ETC, it's going to take the oxygen we've been breathing in, split that apart, and then making water with some sort of the loose hydrogens floating around from our electron carriers. Say that one more time. The oxygen we're breathing in is getting split because H2O, water, only has 1 oxygen. And it's joining up with the free floating hydrogens that were broken off of our electron carriers. And lastly, just exactly like in photosynthesis, that hydrogen gradient, concentration gradient, super high on one side, needs to flow out to help equalize. It wants to equalize that gradient. And as it's flowing through the enzyme ATP synthase, we're creating ATP from ADP. We're adding on a phosphate group to create that triphosphate. And so then it kind of cycles. Some of the hydrogen is leaving through the ATP synthase, and then it's going to get pumped back in through these proteins during the ETC. All right. We're going to repeat it again. Electron carriers, they pass our electrons and hydrogen ions to the ETC. Remember, to trap those electrons, we're sticking on a hydrogen ion to that NAD plus or FAD. And those electrons, when they get to the ETC, will be harvested essentially by breaking off that hydrogen that was holding them together. Those electrons that are released are going to go through the proteins in the membrane. Those electrons have some energy, so they can do the work, that active transport of pumping those H+ ions against the concentration gradient. We're building this concentration gradient. Super important for our last step. At the end of our ETC, so that last protein, we're taking the oxygen we're breathing in, splitting it in half, and putting it together with some of those loose hydrogens that are floating around from our electron carriers to create water. And then our last step, our ATP synthesis, that gradient we've been building, all of that high concentration of hydrogen ions wants to even out. So it's going to flow the only place it can through that ATP synthase to create ATP. Those electrons that we harvested in the Krebs cycle when we created our NADH and FADH2, here's where they come into play. They're super important. They give us the energy to do the work. I'll leave this here for those of you who are doing the notes. The electron carriers are passing electrons and hydrogen ions to the electron transport chain. So we're going from the Krebs cycle to the ETC. The hydrogen is removed from the NADH and FADH2, and so that releases the electrons and those H+ ions. Those electrons go into the proteins to pump those H+ ions across the membrane to create the concentration gradient. At the end of the ETC, that last protein, it's splitting the oxygen that we breathe in half to make water, which is a waste product of this process. The last step, that H+ gradient pushes those hydrogen ions through ATP synthase, our enzyme that creates ATP. Whew, all right. I know that was fast, but please go to column F of our quick write. Answer the question, the ETC uses energy from the electrons to make what? It's making two different things, so give me one of those things. All right. Yes, water is one of those things. That last protein in the ETC is splitting apart our oxygen, combining with those loose hydrogens to create water. The other thing the ETC is doing is it's creating a concentration gradient. The ATP is being made by the ATP synthase, not by the ETC directly. It is helping. It's creating that concentration gradient, which will power the synthase, but more directly, it's creating the water and the concentration gradient of H+ ions. So, great job. You guys are paying attention. Love that. Indirectly, yes, it is creating that ATP. So here we go. Let's count some of these ATPs that we've made. Starting with glycolysis, the very first step, we made 2 ATP. Awesome, on our way. Then we do our pyruvate oxidation, nothing there. Moving on directly, immediately into the Krebs cycle, we make another 2, because remember, there's 1 per cycle. We're doing 2 cycles. Now we're up to 4. Big deal. We need a lot more. Those electron carriers we're making in the Krebs cycle move over to the electron transport chain. This is where the magic happens. Depending on the organism, how efficient their mitochondria are, they can make anywhere from like 30 to 36 ATP. So we can estimate, just for easy numbers, cellular respiration with oxygen, aerobic respiration gives us about 40 ATP for every 1 glucose molecule. That's a lot of energy for 1 glucose molecule. So this is how we get all of our energy. It's all based in this ETC, but we have to get there first. All right, so if you remember from yesterday, we had these questions I wanted you to keep in mind. Why do humans breathe oxygen? What's the point? Why don't we breathe CO2? Well, without O2, that ETC would not be able to produce ATP. Carbon dioxide is a waste product. Obviously, we don't want that, so we're not going to breathe that. But that oxygen that we breathe in helps us create water, which is a product. And some of those hydrogen ions will be used in that process as well. We're also using that oxygen that we breathe in during the Krebs cycle. When we're creating our CO2, we're losing those carbon molecules to create some of our NADHs. And can an organism survive without oxygen? Why or why not? Of course they can. We talked about this yesterday. Anaerobic respiration, no oxygen needed. Bacteria, yeast, some of these prokaryotes, they don't need any oxygen. So why are we learning about cellular respiration and photosynthesis at the same time? Well, the products of one become the reactants of the other. So we have light energy feeding into a chloroplast. That chloroplast produces oxygen and glucose. Then, because that oxygen is present, we're going to go through some cellular respiration, aerobic respiration. We need that oxygen, we need that glucose, we're going to produce chemical energy, ATP. And our products, water and carbon dioxide, will then become important for our chloroplast to make more oxygen and glucose. So it's this connected cycle. We need photosynthesis to survive. Photosynthesis needs us to survive. We're conserving all of this carbon and energy. All right, I know it's been a long period, but one more thing before I let you go. We're going to rewatch this video we started yesterday. As you're watching it, column G, once again, what makes it possible for these plants and animals to live in this sealed glass ecosystem? How are they going to stay alive? Some of you already knew this answer yesterday, but now that we have some new knowledge, hopefully more of you will get this hint. It was the last slide that we just talked about. Declan's got it. The plants produce the necessary reactants that animals need. So that little shrimp needs carbon dioxide from the plant and the sugar that it will produce if it is feeding on the plant. And then the products of that shrimp, the carbon dioxide, will feed the plant. Awesome. Nothing leaves the system. Perfect. We're conserving energy there. Awesome, Ammon. And Kira, awesome. The plant produces the reactants that the fish needs to survive and vice versa. Awesome, awesome, awesome. All right. to stop my recording.